today. Good afternoon. All right, so we're going to get started today. Reminder that this material we start today on vessels is not on your exam on Friday. All right, so just so you are aware of that, this is not on your exam for Friday. All right, so your exam on Friday covers blood and the heart. Okay, so let's get started with this, and let's prevent that from happening anymore so that you don't get sick when you're watching the video later. All right, so today we are going to start on blood vessels. All right, we're going to start. We've talked about the pump of this cardiovascular system, and we've talked about what it pumps through the cardiovascular system. So we talked about the heart as the pump and the blood as the conduit to which, you know, all of this stuff would be pumped through, right? That's what the heart is pumping. But now let's talk about what the blood is being pumped through the vessels. And by themselves, the vessels are considered organs, okay? They are considered organs. They are groups of tissues together that are doing a certain function. All right, so they are considered organs. And there are three distinct types of vessels that we are going to be concerned about: arteries, capillaries, and veins. All right, what do we know about arteries already? Taylor. They go away from the heart. All right, from the heart to capillaries. Okay, in capillaries, what happens in capillaries? Exchange of materials, right? They are small vessels. They're microscopic, porous blood vessels, so you can't even see them. All right, and there's exchange that between blood and tissues, both forward and backwards. Oxygen one way, carbon dioxide the other way, and vice versa in the lungs. And then veins then transport blood from the capillary beds all together back towards the heart, where then it's pumped to the lungs for reoxygenation and exchange. Okay, so general terms: arteries away, capillaries exchange, and then you have your veins towards. They are different in structure. Right, here's a histological picture showing you an artery and a vein. Okay, what you'll notice about these is, let's talk about some vocabulary. All right, there is this area on the inside. No, let's do it this way. On the inside, okay, in here is where the blood is, and that is called what? That's the lumen. All right, the lumen. All right, we have one over here as well. Okay, so the interior, the inside where the blood is the lumen. What do you notice? Which one of these is an artery? Which one's a vein? Do you think? All right, I'm going to tell you that this one with the smaller lumen, okay, and it's constricted and it has a wide amount of a wide vessel thickness. Okay, this over here, wide vessel thickness. Okay, that's an artery. Okay, and then the vein has a thicker lumen. You'll note that the lumen's looks a lot wider. Okay, typically veins have less of a thickness of the wall as well. All right, so there's an artery and a vein that they're kind of juxtaposed next to one another. Okay, comparison of opposites. And you look at this now, and I clear all this out. Okay, zoom in a little bit so you can see. What do you see here? So here, this dark, that's your endothelial lining. right? That's the lining of the lumen. I can go on. And then outside of that, we have the muscles. Okay, there's one layer of muscle. Here's a second layer of muscle. And then outside of that, you have the tunica externa, which is out here. Okay, beyond that, it's out here. Okay? So you can see the three tunics are present in this blood vessel. We could do the same for the veins as well, right? Be a little bit different because of the structure of the vein. Okay? Here's what it looks like in terms of picture. Now there are three layers of tissue that are important in both arteries and veins and in these vessels. All right? We have three layers. We have the tunica intima, tunica media, and the tunica externa. Okay? And they're much different between arteries and veins. Okay? The tunica intima is the most inner layer. Right? It is the inner layer where it's going to be closest to where the blood is. It lines the lumen. Okay? It lines the lumen. And this is where you also find what's called the endothelium, the inner epithelial surface that covers the underlying tissue. Okay? Underneath that, you have a subendothelial layer. Typically, it's like a basement membrane. It's a connective tissue that those endothelial cells will attach to. Okay? That is shared between both veins and arteries. Right? Now, in the middle, the tunica media, that's where we have the big difference. Okay? In arteries, what do you notice about the tunica media in arteries versus veins? It's thicker in arteries. Okay? Within arteries, the tunica media has two layers of smooth muscle. That's where smooth muscle is indicated. All right? And what do we know that, what do blood vessels do that you're familiar with? They constrict and opposite to constrict, dilate. So vasoconstriction and vasodilation. The tunica media allows for that to happen. Okay? And so you have constriction and dilation of these arterial vessels, which will help regulate blood pressure. They will shunt, you'll see it in a second, they will shunt blood away from the surface and through capillaries, etc. All right, so the tunica media, right, and then underneath that is there's a bunch of elastic laminae that was called there. So we have elastic fibers. There's some collagen fibers that are for support. And then we get to the tunica externa. You'll note in veins, and we'll get to veins in a second. Okay? And then the tunica externa is a vasorum. It's the outside. It's like another epithelial lining that's on the outside of a blood vessel. Okay? So if we go back, all right, let's look at the structure of the vein. Tunica intima, still there, single cell layer, right? It's the endothelium. And then if we move out, the tunica media is much smaller, right? Not as many layers of smooth muscle cells, all right? And then the tunica externa has blood vessels. It's a little bit thicker. Tunica externa is a little bit thicker than on arteries. What else do you notice is different between veins and arteries? Nothing. Everything else is the same up there. Check. There's a valve in the vein. Okay? Veins have valves. Okay? Where are veins in relation to the heart? Answer to me all of the vessels that are between the vena cava and the left ventricle. Start with the left ventricle. What do we go into? To the aorta, and then we go into what else? Let's just go to aorta. We'll break it into a bunch of branches and go into the what? More arteries. Okay? Then we go to arteries to what? Get smaller vessels called arterioles. Then we get into capillaries. From capillaries, we're now going to start going back. Venules to veins. Okay? So there's a lot of vessels between the heart, which is pumping blood, and the veins, which still are bringing blood back to the heart. Right? There's a lot of vessels in between there. And some of those vessels are very small, like the capillaries. Okay? And then, so they get very small, and then they start to combine and get bigger. So capillaries are smaller than venules, and venules are smaller than veins. So as you go from large vessel off the heart, they get smaller and smaller, pressure up to capillaries, so pressure gets higher and higher until you get to capillaries, then it goes on the other side, and now it starts to get bigger and bigger, and pressure gets less and less. Right? Where's this blood trying to get back to? The heart. Okay? So the valves do exactly the same thing that the valves in the heart do. What do the valves in the heart do what? Prevent backflow. Okay? And the valves do the same thing. These are structured similarly to the semilunar valves that you have in the aorta and in the pulmonary trunk. So as blood is being pushed through here, and a lot of your skeletal muscles work when they move and they contract, they will milk blood back through the veins. They'll milk it through, and then eventually your skeletal muscle relaxes, and it relaxes the pressure on the vein, and then the blood would start to flow backwards. Well, it fills that valve and prevents it from going too far. And in some cases, those valves fail. When those valves fail, you get painful pooling of veins. Does anyone know what that happens?
Varicose veins.
Varicose veins, especially in the legs. Okay, because it's also fighting gravity. Okay, so the valves are important. Okay? So, comparison-wise, arteries have thicker walls, they have thicker tunica media and a narrow lumen, right? More elastic and collagen fibers. Why do they need to have a thicker wall and more elastic collagen fibers? Think about this is about relationship of structure and function. Their structure is related to their function. So they have thicker walls. Why? Emma. They're going to pump oxygenated through. Well, we're going to transport oxygenated blood throughout the body. What about? I'll give you an analogy. Okay. You all know about how thick a garden hose is, right? They sell them in different diameters, but the actual rubber thickness is, it's a garden hose. It's not going to go in, something you want to hang off a building from, right? So now say I take that garden hose and there's a cap at the end so no water's going to escape, and I attach that garden hose to a couple different fittings and then to a fire hydrant. Now you know about fire hydrants. What's the pressure inside fire hydrants? The amount of water that comes out of there. A lot. Okay? So now I hook it up to a fire hydrant and I turn it on, very low. What happens to the garden hose? Now there's no way for it to leave, so that it's a fixed volume. What's going to happen? Water's going to come out. Fire hydrant. What's going to happen to the garden hose? It's going to explode. Okay? It's going to explode. Why did it explode? Too much pressure. Okay? But if I had a same diameter of the garden hose, the inside, but a really thick hose on the outside, right? And it was still gonna, so the volume of the hose is the same inside, but I have a really thick wall of it, right? So I have a garden hose with this much thickness. Still the same amount of water that's gonna go in there volume-wise. What's gonna happen to the hose now? Do the same thing, hook it up to the fire hydrant, what happens? Same amount of pressure. Does the garden hose break? No, it doesn't. Well, at least it takes more pressure to do so, right? Same thing is happening here, okay? The arteries and arterioles are subjected to higher pressure than venules and veins. So they need a thicker wall, okay? Or else things happen. And some bad things that can happen when a blood vessel wall bulges out or breaks, okay? Now, the veins have a thicker tunica externa, larger lumen, right? Thus they have a lower pressure. And then your capillaries, okay? They don't contain the tunica media, okay? Only the tunica intima. So they're only very few cell layers thick, right? There's only the endothelial cells and basically a basement membrane. Why is that important? If we only have one cell layer thick, why is that important, Emma? Diffusion. Exchange is going to happen here. Things are going to move from one side to the other. If we have a larger wall, that's going to prevent the movement of materials. Okay? Thin wall allows for rapid gas exchange and nutrient exchange, right? We want that to happen as fast as possible. Because blood is always going to be moving. So if it moves fast enough, right, or if it moves slow enough for something to happen, we still want it to happen fast enough so we can have exchange occur at its best rate. Okay? So we can look at the differences again, and this I thought was a great picture that's in your book or in your text about how things are. Okay? And we're going to talk about large veins versus venules, right? And we're going to break down arteries versus different types of arteries. What you'll see in all the arteries on the right-hand side is that they have a thicker tunica media. Okay? So those three layers, tunica intima, tunica media, tunica externa. All right? In arteries and arterial blood vessels, the middle, the tunica media is thicker. Okay? So, arteries, they branch. When from your dissections and your lab on the heart last week, what did you see? So, one blood vessel, two blood vessels come out of the heart, right, that are flowing blood. One from the right ventricle, one from the left ventricle, right? And then what do? The aorta is one, and then what happens at the aortic arch? It branches, okay? So we're not just going to have one vessel, we're going to branch. And every time the arteries branch, it gets somewhat thinner in diameter. The lumen gets smaller in diameter, all right? As it gets smaller, it helps maintain the pressure. Okay, we're going to talk about a couple different things in this chapter that I want to go over right now. Okay, it's a relationship between pressure. Let's erase that green color. Pressure, diameter of a vessel, and flow. Blood flow. Okay? Blood pressure and the pressure and the velocity of flow, okay, are a function of the diameter of the vessel. Okay? So, what it looks like is this. You have a blood vessel, right, and you have a diameter across. Okay? I'll make it bigger. A diameter across that blood vessel. Okay? So this would be blood vessel number 1 over here. Now, you can control the diameter of that blood vessel. You can make it smaller or larger. So we have smaller versus larger. Okay? The larger the diameter, the less pressure, okay, and the higher the velocity of flow. So if you have this vessel here, let's say vessel number 2, okay, that's a 3. All right, so vessel 1, its lumen is very small, okay? Now an amount of blood is pumped through here. What do we call the amount of blood that's pumped out of the heart per stroke? Stroke volume. Okay? Now, it's a fixed system. Ideally there's no end opening. It's not an open system. Okay? It's a fixed capacity of blood that we have, right? We can produce more, but we lose some, but you have, on average 5 to 6 liters of blood in you. Okay? Hopefully you're sitting there not losing, hemorrhaging blood. If you were, let me know, raise your hand, we'll take care of it. Okay? So your heart is pumping out blood into a closed system. All right? Now, if you take those vessels, that system, and reduce the diameter of all those blood vessels, you reduce the capacity of the system. You reduce how much volume can be there. Right? That increases pressure within the system. Okay? Now, the blood vessels, if your heart goes up, it's not going to, your heartbeat goes up, increased stroke volume, your blood vessels aren't just going to start popping open, right? They can withstand a certain pressure range. Okay? So the relationship is that as this changes, you have vasodilation and vasoconstriction. It changes the diameter of the vessels, which lead to changes in pressure and flow, velocity of flow. Okay? Where do we want the velocity or how fast blood is moving through the vessels to be the slowest? In capillaries. All right? We want capillaries to go slow so that exchange can occur as fast as possible. Right? We get efficient exchange. Okay? So we want that to be the slowest. All right? Think about as we move forward. Let's talk about the different types of arteries. Okay? Here's an elastic artery. They're your largest arteries. Okay? You can see the tunica intima, a large tunica media with elastic fibers throughout. All right? And then the tunica externa on the outside. A large wall, elastic fibers. What are elastic fibers going to do? Allow for? Think about the aorta. The aorta is an elastic artery. What does the aorta do every time the heart beats? It gets blood from the left ventricle. Does it stretch a little bit? Yeah, it does. Okay, so a little bit of stretch. So this large diameter, right, it conducts blood away from there. So you have the pulmonary trunk, the common carotid, and common iliac arteries. All of you, take your right hand, place it just on the side of your Adam's apple, and you should be able to feel something right there. What is that? Your carotid artery, and what is it? What are you feeling? Your heartbeat, which is your pulse. Mechanically, what is happening? What are you feeling right there? You're feeling blood go through that blood vessel, right? And if that blood vessel didn't stretch, would you feel it? No. Right? So it stretches. There's a wave of blood that's pumped out of the heart every heartbeat, every contraction that pushes blood along. Okay? That the vessels stretch a little bit every time. If they didn't have elasticity, they wouldn't go back, and you wouldn't be able to feel that pulse. Okay? All right, and some other examples: common carotid, common iliac arteries, the femoral artery as well. Okay? So stretch and recoil. And that stretch and recoil actually helps push blood through the system. It aids the heart, so it's not just doing it all on its own. Okay? Muscular or distributing arteries. They're medium. Okay? Distribute blood to specific regions, right? And you have muscle that allows for vasoconstriction and dilation. All right? You have two layers of elastic tissue in this, internal and external. All right? So coronary arteries in the heart, brachial artery in the arm, right? You can go from here. These are the ones you're most commonly going to see in the lab, right? Probably in lab next week. Questions? All right, still thick tunica media, okay, with two layers of elastic lamina top, internal and external. And then we get smaller. We have arterioles. Okay? Arterioles. These are the most important vessels that help maintain and regulate blood pressure. Okay? The smooth muscle of arterioles are usually what we have display a vasomotor tone. Okay, where the smooth muscle is somewhat constricted, okay, to help maintain blood pressure. You change that, and it becomes too dilated, and you get deregulation of blood pressure, right? But they regulate pressure and blood flow, right? So the arterioles are your most important factor. And the dilation of arterioles are your most important factor in regulating blood pressure. So let me ask you, someone who goes into the doctor and has a blood pressure reading and is diagnosed as hypertensive, having high blood pressure, okay? If the doctor could take a measurement of the diameter of the arterioles, okay, before or at diagnosis, and then after that patient goes home and on a low salt diet for 2 weeks, which one, before or after, would you have a larger diameter of arterioles? What do you think? What's going to happen to blood pressure if someone goes off of goes on a low sodium diet for 2 weeks? It's going to go up or down? Down. Okay? And that's going to be what's going to happen. If blood pressure goes down, what happens to the diameter of arterioles? It's going to get wider, right? They're going to dilate. Okay? In some cases, we have problems with blood vessels. One of the biggest ones is atherosclerosis. Okay? And this is a buildup of plaques or atheromas, right? And this is a thickening of the tunica intima, okay? And a narrowing of the arterial lumen. So what you see here in this picture, all right, you have this right here is the lumen of the vessel that's left over. This is the whole plaque. Okay? It has started and then been repaired and smooth muscle has grown over it. What do you know, what do you say about the size of this, relative size of this vessel? The diameter used to be this. Right? Well, let's maybe a little bit less than that. The diameter used to be about there, and now it's been reduced to this. Okay? What happens to the pressure within that vessel? It increases. How much? A lot. Okay? So the pressure within there's a mathematical relationship with the increase in pressure to the decrease in diameter, right? And if you reduce the diameter by half, you increase the pressure by 4. Okay? So pressure goes way up. All right? And this could be due to infection, inflammation, plaques, and you can have high cholesterol. This could be a fat plaque. The plaques don't necessarily have to be just fat, right? So this is what we call a hardening of the arteries, right? And they lose their elasticity, okay? And narrowing that occurs, okay? What does that lead to? What does this directly lead to in many cases? Heart attack, all right? So increased cholesterol in the blood, right, makes you prone to disease. Males more affected than females, and smoking and hypertension, right, and diabetes are direct risk factors, okay? So what do you do to, now my father-in-law had a heart attack 12 years ago. 12 years ago. And he had a coronary heart attack, myocardial infarction. And then he had to have a stent put in. Okay? This is what a stent is. They treat these atherosclerosis, this atheroma, with what we call angioplasty. Okay? So he had a coronary clot that had moved. So not only do you have an increase in pressure where you have a disruption in the diameter, but what happens when that plaque piece of that breaks off and moves from one place to another, and it happens, and then clogs the blood vessel. Now you have a clot that forms, and now cuts off a portion of the coronary artery, or at least reduces the capacity for for blood to flow through, so you need to open it back up. Okay? So cholesterol builds up, partially blocking the artery. So they take a catheter, put it through your femoral artery, up the aorta, into your heart, or wherever the blockage is, and they open this up. It's like a little balloon. And they spread open the vessel. Right? And in this case, with the stent, they have this wire mesh that's open, and it holds open the vessel. Okay? Now blood can flow through more normally from the expanded stent, returning blood flow to the heart or to wherever it's going. Okay? In extreme cases, you could have what's called a coronary bypass surgery, where they'll crack your chest open and reroute the artery around where the clot is, or around where the plaque is, so that blood can flow normally through that, right? Or they'll take a piece of femoral artery and they'll put it in your heart and kind of loop that over, and they do that as well. All right, kind of bypass the coronary surgery. All right. Another problem with blood vessels. All right, if pressure's too high, in some cases, the vessel wall can balloon out. So if you have a vessel wall that looks like this, right? What I mean by balloon out, that's the normal vessel, right? But if you end up with an area of it that balloons out, because of pressure, because it forces it open, okay, what ends up happening? Well, blood pools in here, okay, and that wall along the ballooned area becomes more weak. It's stretched, so it's a thinner layer. And then pressure continues, and it cuts, or some type of trauma, injury, blunt trauma to that causes it to open, and you end up with bleeding that occurs. Okay? It happens in elastic and muscular arteries. Okay? It is common in the aorta or arteries at the base of the brain. End up with something looks like that. All right? So this is a brain looking at what's called a hemangiogram, looking at a blood vessel diagram showing blood coming into the carotid artery going around, and look at this right here. By radiograph, what does that tell you? Again, the brain and inside the skull, that's a fixed volume. What do you think that blood vessel just did? I put a lot of pressure on the internal surface in that, right? That could cause, if that aneurysm ruptures, that's going to cause death. Okay? Lots of problems with that. Questions on blood vessel, the walls of blood vessels? All right, let's talk about capillaries. Capillary characteristics, they're small vessels. They are, diameter-wise, the smallest ones are about the width or about the diameter of a red blood cell. Okay? So that red blood cells can freely move through. They're flexible, so they can kind of go through easily, but they make them stack, so they travel in a single file line. Here's the blood vessel. Right? Here's your red blood cells in a stack, so they kind of go through in a single file line. Optimal, thin wall, small diameter, slows the flow of blood cells through the vessel, allows for optimal exchange. Right? Structure, function, relationship. This is where exchange is going to occur. You want the blood cells to go through slowly, and we want as little barrier as possible between the blood cells and the surrounding tissue. There are 3 different types of capillaries. Okay? Continuous, fenestrated, and sinusoid. All right? Continuous ones are what we're talking about here. Okay? They're just going to run through, nothing big. Okay? The endothelium is continuous. Okay? Tight junctions connect cells. They don't form a complete seal, though. All right? So here's your endothelium, red blood cell within it. All right? And now you can have exchange that occurs, and on the outside of the endothelium, you have the basement membrane, which they're attached to. Okay? Found in the skin, the lungs, muscle, they're very common. Okay? Some things can pass through, pretty small, glucose, right? Some tissue, fluid, right? But not the formed elements of blood, right? Unless a white blood cell squeezes through it somehow. Okay? Cells and proteins are stuck in the blood. Those are continuous capillaries. We have fenestrated capillaries. Fenestrated. They have what we call fenestrations, openings, where the tight junctions aren't complete. Okay? And fluid can flow out a little bit. There's pores, that's what they're called. So you see, it basically looks like the structure of the continuous capillary, but he was the guy who had to kill himself with the fork to not make a good canoe for the Native Americans, right? So he had to punch holes in it so that blood could flow out, okay? Think about where these are, what they do. Those extra pores are going to do what? They're going to allow for what to happen? Not just exchange of gases, but what's going to happen when bigger things are going to move through. Okay? More things, more holes, more openings. Not only gas exchange, but other things are going to move through. And so you have, this is where absorption of nutrients is important in the intestine, the stomach. Okay? Where filtering occurs, right? The capillaries of the kidney, where we're going to take substances out of the blood or put them back in. So we have to have a way of transporting them in across the endothelium a little bit easier because there's a little bit more stuff there. Okay? So that's where they are. So absorbing nutrients and filtering blood through urine, fenestrations. And then finally we have sinusoids or discontinuous capillaries. All right? The endothelial cells are even more incomplete. There's no holes, but they're just large gaps, large separations. All right, so a couple cells are attached, and then there's a big open space. Okay? So that open space allows bigger things and allows the blood to pool, right? Red blood cells kind of pool in an area. Okay? And this is where in blood, you have this in bone marrow, and you have this in the spleen, in the liver. You have these sinusoids. Blood kind of pools, and this also allows for areas where you have white blood cells, and you're going to clean out those dead blood cells. All right? Or you're going to produce, like in the case of the bone marrow, produce new blood cells that will take place. All right? So you have a discontinuous basement membrane, those large openings, and blood can kind of pool in an area. So different types of capillaries. Continuous, fenestrated, sinusoidal. Right? Difference in structure, difference in function. Okay? Let's talk about capillary beds, because this is where the action happens. Okay? Blood flow through a capillary. So blood is going to flow from an artery, all right, to an elastic artery, to a muscular artery, to an arteriole. Eventually, there's a meta-arteriole that feeds into a network of capillaries. And along these network of capillaries, that's where exchange is going to occur. Okay? So imagine that this is embedded within a tissue, and there's all tissue cells around it. Along those capillaries, blood is going to divert off of the middle arteriole into different capillaries, right? Maybe they are what we call it true capillaries, all right? And they're open, that allows blood flow through and exchange is occurring. That's why you're going from red on the left here all the way over to blue, where deoxygenated blood is going to start to return back to venules, eventually to veins. All right? That's the way blood flows. Artery, arteriole, through the capillary beds, eventually to venules and veins where they collect back. All right? Now you'll notice here there are more than one way to get from arteriole to venule. Okay? There's a bunch of side streets that you could take, or you could take them one main road all the way over. What's the benefit of that? What happens if you go and you take a couple of these side streets? You take this route here as opposed to this route. How fast does it take? Casey? Takes longer to take those side streets, right? Okay, but you get to see better sights. You get to see the different neighborhoods that are there rather than going up the main road, right? But sometimes you want to go from arterial to venule. Sometimes you want to cut this off and stop capillary exchange and keep things for yourself. So these little muscles right here, we call precapillary sphincters, they can shut off. They can close these roads. Okay, they can close these roads right here. If they close the roads, what happens to blood? It goes straight through. And now no more blood is out through here, just going straight. Okay? That's what we call shunting. Okay? So when your capillaries in your fingers, all right, from playing out in the snow, your hands get a little white, right? Because it's cold, this is what's happening. It's preventing heat loss by shunting capillary movement, right? And this is a continuous capillary, right? While these are, continuous, they could be fenestrated as well. Okay, but you can cut off the sphincter so that blood doesn't flow through and eventually just goes right back to the venule system, the venous line. Okay, veins or venules, smallest veins. All right, they're in the post-capillary venules, the largest venules having 3 tunics. They merge to form veins. We already talked about the fact that veins have valves, all right, which prevent blood from pooling. All right, we will talk about how they get back to, we'll talk about systemic, actually we could do this one right here, okay? The venous blood, or the venous circulation, is actually a reservoir, all right? At rest, 70% of your blood is in the systemic circulation, all right? So 70% systemic circulation, you have 12% in your heart and 18 in the lungs at rest. Okay, we still have to have a lot in the lungs so that we can oxygenate blood, but all of it, most of it is in the systemic circulation. Now you wake up and in the morning you go for a run. What happens? In the middle of your run, now what happens? What happens to these percentages? How do we get blood to go from the venous side to pumping around all? Now we have to redeliver stuff, right? Systemic arteries only has 10%. Systemic veins has 55% of your blood at rest. Does that percentage change drastically when you exercise? Yes, it does. How does it happen though? What happens? Yes. Increased heart rate. Yes. What else? Think about Taylor. We're going to increase oxygen intake, so that means we have increased heart rate, increased oxygen intake. Casey. Stroke volume is going to go up. Our cardiac output's going to go up. Heart rate's going to pump, and now it's going to move blood from here, from the lungs. We're going to start pulling stuff out of the reservoir into our tissues are demanding more and more nutrients. Okay, more oxygen, more glucose. So we have to give it to them. All right, so we have to pull this out. When we pull this out, what happens to your blood vessels? What happens to your blood vessels when you exercise? What happens to your skin? It dilates, right? Your skin gets red. Blood vessels dilate. The arterioles dilate. Your blood pressure goes up really high because your heart rate goes up. So your arterioles have to dilate, and now it moves all of this blood onto the systemic side. Onto the pulmonary or the systemic side. Out of the veins though, and into the arterioles, your blood pressure goes up, and your heart compensates. All right, we'll pick up this on Wednesday. I will post your Connect assignment today, right after this class. Okay? It is not due until Wednesday of next week. Okay? You can use it to study for your exam on Friday. Right? But it's not due until Wednesday of next week.
I've been trying to email you last week.
Yep.